Good morning, everybody. I am Maria Letizia Barreca. I am a professor of medicinal chemistry at the University of Perugia in Italy. Today, I'm going to provide you a brief overview of our project that this year was founded by CJD Foundation. Just a quick introduction. It is well known that the key event for the development of prion diseases is the conversion of a normal physiological cellular protein called prion protein, PRPC, into a pathological form that is called PRP scrapie. PRP scrapie then accumulates in the brain of affected patients, leading to neurodegeneration. However, PRP scrapie is not the only cause of these devastating disorders. There are strong evidence that also the normal protein, the PRPC, has some key role in this development of the prion diseases. In particular, PRPC is very important for PRPC propagation, so we can have a larger amount of the pathological form. But what is more important to us is that PRP scrapie needs the normal protein, so needs PRPC to give neurotoxicity. And so in the last years, we have worked in a project with the idea to develop compounds able to suppress, to block the PRPC-mediated toxicity. In particular, about 5 years ago, we have identified a compound that showed some promise in cellular models. Later, we started, to work on this compound to put much efforts, and we have designed and synthesized about 100 molecules. The testing of these molecules led to the identification of some compounds that were very potent in a cellular model of prion diseases. And in addition, these compounds also showed some, I would say, suitable physical-chemical properties for in vivo experiments. And here we are with the CJD Foundation project. So we asked the support to test the efficacy of these compounds in an animal model of prion diseases. In reality, we already had in our hands, a mouse model of genetic prion diseases, but we need to validate the model. And this is what we have done in the first part of the project. At the same time, in order to test compounds, you need large amounts. So we have also synthesized the required amounts, and we have the compounds ready. So we already achieved this second task. So you know, at this point, we have the model, we have the compounds, so we are ready for the in vivo experiments. We already asked for some permission because you need some special permission for in vivo experiments. And we are confident that we will start this kind of experiments in autumn. At that point, we will have a proof about the efficacy and also the benefits that our compounds can provide to prion diseases. You can understand very well, I guess, that this is a very complex and multidisciplinary project where we need different expertise and competence. And so I would like to acknowledge my collaborators. At the same time, I really would like all the donors that supported our project, and of course also the CJD Foundation. And thank you all for your attention.